Good morning, everyone. It gives me great pleasure to welcome everybody to the conference today and introduce Dr. Chamala, who is our speaker for today. Dr. Chamala received his bachelor degree in science and bioinformatics from Brigham Young University in Provo and followed it with Master of Science in Bioinformatics at the University of Illinois Urbana-Champaign, and subsequently received his PhD in botany and plant biology at the University of Florida, Gainesville, where he continued his academic career and transitioned to pathology, immunology, and laboratory medicine as a clinical assistant professor. And right now, he is associate professor of clinical pathology at the Department of Pathology, University of Southern California in Los Angeles, as well as a director for Center for Pathology, Informatics, and Data Science at Children's Hospital of Los Angeles. So he had a very early morning to join us, and I definitely appreciate that. And he is an author of close to 50 publications and served as a PI and co-investigator on several NIH and externally funded projects. And today he's going to talk to us about the precision genomics medicine challenges, opportunities, and the roadmap for effective delivery. So the floor is yours, and thank you for joining us today.
Yeah, thank you, Dr. Mroz, and thank you all for joining this early morning presentation. And yeah, so today I'm going to precisely talk on like precision genomic medicine challenges, opportunity, and roadmap for effective delivery. Again, this also sets out a good timing for your department where you're trying to get into EPIC genomic module and and also EPIC Beaker genomic module. So this will be perfect timing for the implementation, and we have a lot of experience in this area. I'm going to share some of the experiences what we had. So overall, the objectives is to we basically go through the illustration of current environment for delivering precision genomic data for clinical care and also for translational research. And I'll summarize challenges and opportunities in managing precision genomic data and outline for the roadmap in developing genomic data exchange standards and interoperability. So I'm going to start off my presentation introducing briefly about precision medicine. I think many of may already know, but for some of you who are not into molecular pathology or anatomic pathology, may be new. And I'll just give you an overview of clinical genomics and how currently clinical genomics data is dealt in LIS and also EHR systems. And finally, I'll conclude my talk going through some of the national and international efforts for genomics data standards and exchange. So I think the traditional medicine has been, at least, where one size fits all. It's almost like you go with certain symptoms or something, doctor, and everybody gets the same medication or something. But as we know, not all the time the medication or the prescription will work. We see some individuals having some adverse reactions. Some of them may not have any benefit, and some of them have benefit. Many of them would have benefit. So we know that the one size fits all approach is not quite successful approach. So what precision medicine try to do is it's one of the ways, right? And precision medicine has multiple aspects. One of the aspect is how do we, tailor, for example, in this example, medication, based on the genetic profile. So based on translational research, people get to know that, okay, individuals with this genetic profile, will have adverse reaction, and some of them with some such genetic profile have benefits from the treatment, and some of them, always have beneficial treatment. So based on the genetic profile, basically we tailor our treatment plan for the patients. So I think this is precision medicine, but I think oftentimes there's also confusion about what precision medicine is, right? Basically people think for individual person, but it's mostly for a group of people. It's not, each person gets really tailored thing, but it's more of a group of individuals with similar genetic profiles. Okay. Um, and uh, and also it takes account into genetics is only one portion of it, of precision medicine, and oftentimes now almost precision medicine became synonymous for what do you call precision genomic medicine, if you think of. So genetics is one aspect, environment is their lifestyle. There are multiple aspects where you could tailor the treatment plan. Again, this is, again, as I mentioned earlier, it is for groups of patients, not for each individual nuclei. So there is some differentiation you should understand. So this is one. And again, the term precision medicine could be newer one, or at least in last decade or more than that, but it's more buzzword. But the concept was not real. For example, a blood transfusion, we do based on the matching. It's more tailored for the individual, for individual group of individuals with certain blood types and so forth. So the concept is not new, but the term and the prevalence of the term is quite common nowadays. So overview of clinical genomics. So this is the minimal need kind of thing within an institution, right? So you have a clinician, or most of the clinicians, or sometimes pathologists, they order genomic tests. So again, I'm going to present some high-level challenges and opportunities here in this slide here. It's a very high-level overview. So genetic tests, compared to other testing, right, other pathology testing, what we order, for example, chemistry tests or others, there is a lot of variety in genomic tests, and it's a lot of confusion. Okay, should we order next generation sequencing tests? Should we order array-based tests, or maybe PCR-based, single analyte tests? So there is a lot of variety of tests, and which is also confusing subject for ordering providers. Okay. And the second, once the order is placed, it goes to pathology lab, right? It could be your local lab with in-house lab, or it could go to external labs like Foundation Medicine or Tempus and some other labs externally. Okay. So the thing is, another big confusion here, right? For example, if you order CBC, to LabCorp, or you order CBC to your local lab, the content of the test, maybe 10 or 12, whatever the result components, going to remain pretty much same, right? Or similar. Maybe there could be really minor differences, but not too much. Whereas in molecular testing or genomic testing, clinical genomic testing, it is very variable, in the sense that the content, for example, if I order genetic test, cancer testing, somatic testing to University of Minnesota versus maybe Children's Hospital, let's say, or University of Florida, they would have a different gene content. And one of them may have test for 500 cancer genes, some of them may test for 200 cancer genes. So it's a lot of variation among the gene content or underlying testing is being done. And also there's a lot of variability in terms of bioinformatics pipelines, and also assay specificity sensitivities, and also different reporting styles. So it just, the whole point here is there's a lot of variability in where you order, what you get back, and so forth. So there's another challenge for providers to really understand what they're getting into. And the results are basically in the lab, or finalized and verified and finalized by the pathologist. And the other, for the molecular pathologists, one of the challenge here in the lab is, so when you, for example, if you sequence the whole genome sequencing, let's assume, right? That's a really extreme example, or this is very much reality right now. A lot of whole genome sequencing is going down right now. So you would end up having a few million variants, genetic variants, for each individual for whole genome sequencing. There's a big challenge for the molecular pathologist to basically getting those 2 million variants into maybe 5 or 6 or even less than 10 if you think of variants to report, which are kind of actionable or close to actionable. So again, that is another major challenge in this field. And the next step, once the pathologist signed out the report, is results are get into electronic health record. As I mentioned, only actionable variants go into EHR, not everything, not the raw data. The advantage of that is the physicians, can focus on the patient care, not going through all the 2 million variants and understanding what they need to pick for the disease they're trying to treat. So that advantage. The disadvantage is I think some of the clinicians tend to be what you call more translation research focused, or they understand the disease very well, understand more genes than what a pathologist would understand, or even the software we use for interpretation would understand. So there's a loss of that. If you have a raw data, some physicians could make better decisions, could be, again, potentially, but also in the future there's some clinical trials based on some molecular profiles that you cannot tell because the data is not in EHR. You only have actionable variants, so that cannot be done. And also for translational research, the data again sits in the pathology lab, not within the EHR. So the researchers do not have access to the data in the EHR, so they cannot really use the non-actionable variants for their future translation research. So once the results are sent to electronic health record, a lot of times the scanned image are discrete data fields. I'll talk about that more in my presentation. And the physicians, view the clinical report, again, the only actionable genetic variants, and make the decision, based on the report. And there's also a trend to put the results, obtained into a genetic repository. For example, at University of Florida, before coming to CHLA, we developed a DNA genetic variant data repository, and people could use simple things, right? CBio portal and other things for storing your genetic testing data locally. So anyway, so this is one of the trend you can do. But this is something, a minimal needed kind of thing, but there is a robust need would be, hey, when we're doing this, performing lab, for example, if you're doing it in your local lab, can we also store the raw genomic data, meaning and also some of the intermediate files? Because the reason for that is, in the future, if you want to, there's a newer bioinformatics technologies comes out, right? You could go back and reanalyze the raw data to get more accurate insights or accurate analysis. And also having those raw data, for example, will also give access to your translation research at the institutional level to perform their research, instead of collecting the samples again and sequencing them. Okay. Again, this definitely, even for the private vendors, for example, like I was talking about Foundation Medicine or some other companies like Tempus or others, there could be a university-wide agreement with the vendors to give the raw data back to the university. So that's always beneficial. But again, there are challenges I want to get into a little later. So, and again, not just getting the raw data, but also being able to, store the data in a repository, and being able to make, for example, you have all the variant data in a repository, right? So then in the future, there is a new therapy available for certain mutations, So currently, it may say there is no therapy available for that mutation, but in the future, something comes up, then in a real time or something where it can alert a clinician or a pathologist saying that, hey, this patient got a new therapy, a new clinical trial available, would you like to enroll or prescribe something? So that will definitely help for the clinical care. And the last portion is also having this robust genomic data repository helps with the IRB approval and again developing this honest brokering process and so forth. So I think again the star ratings I'm giving here is more my personal observation. It is not a published study. I just want to disclose that. I think community gonna agree mostly what I'm saying here. This is a very generic at this point. The sequencing costs, has dramatically dropped down. I think maybe when whole genome sequencing of humans started maybe over $2 billion, and now at this day and age, maybe a few $100 you can do whole genome resequencing. So which is quite cheap. It's not that expensive, and things gonna drop down quite a bit also in the future with a lot of competition coming in. So I would say this is not too much of a barrier at this point. Okay. And analysis pipeline development, right? The bioinformatics, I think if you ask me, 5-6 years back, bioinformatics analysis was a little complicated because you have to, develop your own analysis pipeline on your local systems or on the cloud, whatever it is. You have to develop your own. But right now there are, for example, Illumina has their own the Dragon pipeline, and everything is almost a click button kind of things. People can go through. You need not be a bioinformatician to do those kind of things. And there are a lot of other companies also are offering a pre-built software for analysis of pipeline, bioinformatics genomic analysis pipelines. Again, which significant progress has been made, and again, some gaps need to be filled by internal bioinformatics team, but mostly things have been pretty standardized. And analysis speed, I'm giving 5.0 for it because, as you know, the cloud computing, we have AWS, Microsoft Azure, and Google, and Oracle. So speed is not an issue. If you have resources, right, you could do whole genome analysis less than 20 minutes. It's not a big deal, right? You can ad hoc wise, you can just request resources, how much ever you need to make analysis faster. So this is 5.0. Very, very much progress is made in this area. But one of the, I think, challenging areas, still a lot of progress need to be made is, I was talking earlier about, I'm giving a whole genome sequencing an example because it's a little bit extreme example.
Me, I was driving and this crazy car on my right hand side.
Hey, Tony, can you mute?
Yeah.
Okay, so the analysis, so I was mentioning earlier, you have for whole genome sequencing, there could be 2 million plus genetic variants. Pathologists need to filter them out into more actionable or relevant to the disease variants, right? So for that, it's still a process. The tools are there, but it's a lot of time people are going to spend, especially if you do germline kind of a NICU kind of setting. It is a lot of work, and people spend, especially the molecular pathologists and the genetic genome analysts and stuff. They spend quite a bit of time in doing the interpretation. So that is still a lot of effort need to be done in terms of technology to get the time down on that aspect. And I think this is I gave a really poor rating for data integration and interoperability. So this is a key aspect of a clinical or precision genomic medicine right now. Is as we're generating all this data, but how are we exchanging the data in electronic format more in an interoperable manner, right? Do we have a standards to exchange the data? For example, I'm just going back to CBC example, right? It's a no-brainer, like everything is automated, right? When the machine does the testing, it just automatically interfaces across EHR systems, not only within the institution but also across institutions. So it's pretty seamless. There's no clicking buttons there anymore. But for genomic testing, that's not the case. It's mostly the practice is you do exchange data using PDFs, which is not such a good way to do it. Again, I'm going to talk more about the current state of this thing. So today's talk, would be mostly focused on this data integration and interoperability. So clinical genomics data in LIS, I think I'm going to talk maybe. I spent significant of time on understanding current practice and challenges. So what does a genome can tell physicians? We know that it can tell diagnosis in some aspects, prognosis, and treatment plans for the physicians, using the genomic testing. The challenges of representing genomic variants in the EHR are several. One of them is, there are several hundreds or millions of variants, you get when you sequence or when you, do the genetic testing for a patient. Now, how do you deal with those things in the EHR? EHR is not really meant for, storing millions of, result components for a single test, right? So that is not really, compatible system in that sense. It's not designed for that. And evolving clinical interpretation guidelines and variant reclassification. If you think of it, there are a lot of FDA approved drugs based on the genetic profiles, and guidelines has been changing. So how can you, and the EHR are not really designed to have those guidelines updated, or it's not their responsibility. So it's another challenge there. And especially variant reclassification, there's another thing, right? You already have variant data there. For example, pharmacogenomics, you can take an example. We already have some current CP guidelines now, but in the future, there's new guidelines. And again, you can update it, but again, that is not the EHR driven thing. It's more, it should be done some way of some curation from the institution itself. And as I mentioned earlier, there's a lack of evolved standards and adaptability by EHR and LIS vendors for data exchange. One of the major hurdle also still is many clinicians are not adequately trained to manage genetic testing results because of this complexity I discussed earlier in one of the slides. So current and future practices, right? Current practice, most of the practice is reported as a PDF, the genetic test, my previous institution, I developed some, clinical lab value based approaches for storing the variants. It's similar to clinical chemistry tests, but again, and lately, we moved to Epic EHR vendor native way of storing genetic variants. So I'm going to explain this more detail in the coming slides. So again, this is a very common practice for the storing genetic test reports. The advantages here is, again, it's very easy. I mean, it's a common practice. It's very common practice, so it doesn't require much training, right? We know scanning, reports is pretty common in the clinical setting. So it's very easy. Also, it's very easy to enter. You're not really entering the data. You're just scanning the data. There is less chance for error prone. But obviously, the limitations are you cannot access the data. Difficult to access within the EHR because they're buried under several other scan reports. So it's very difficult. And also, you cannot search. This is not searchable because these are images or PDF files. So if you want to do a retrospective analysis or reinterpretation, I think many of you may already have experiences. You ask your resident or a graduate, student or a fellow to basically go to EHR and just download everything manually and take a note of your variants manually also many times. If you have this a scanned image, the challenge is you cannot really do a clinical decision support because you can do based on the discrete data. So you can write rules around discrete data, but without which you cannot really do it. So at University of Florida, my former institution, back in 2016 or around 15, I think I came up with some idea about this idea about how could we store genetic variants as a discrete values. So because EHR vendors were not supporting, right? Having a native support for this. The way we did was, similar to the chemistry test, or I'll go back to the CBC example, right? It's a very simple exercise test. So CBC is a panel. You have multiple result components, and if you think of genomic testing, is also kind of is a panel, right? Nowadays, right? You don't do single analyte testing, so often. So you have in genetic testing, you have multiple genes, each gene winning, one result component. If you think of hypothetically, okay. So it's kind of a panel testing. And so in CBC, you would have white blood count, red blood cell count, or whatever the counts you're going to have here. Instead of that, you're going to have a result components would be the genes, and the value would be the genetic variant. Okay. So for example, in this case, if we had 20,000 genes, okay. If we have mostly when we started off. We started our somatic panel with using, I don't know, less than 50 or less than 100 genes, So we are able to build those result components back then, and the likelihood, most likely, we get one very actionable variant for each gene. So we are able to represent similar to our chemistry test, and once this is how we represent, and once this is an EPIC beaker. And once you sign off, this is how it shows up in the EPIC EHR view. Okay, so you have pathology, you have a molecular, and you have a genomic is a NGS panel there. So where it shows you only the genes names where you found the variant, right? So this is how it shows. And when you, for example, we have done this testing for this patient back in 2019, in April. And there are no variants detected, but later in the May, we found some genetic variants, and then we are able to show here. And when you click on this link here, it also opens the full report in a PDF. So this is far better, right? Now we went from a PDF to more a searchable manner, right? In a discrete manner. We can pull the data easily. We can, showcase the data very easily on the patient result review section. And not only that, you could retrospectively analyze this data or even build a clinical decision support around this. Okay. So the advantages I already mentioned: retrospect searchable and easy accessible for the clinicians. The limitations is, imagine, again, we started off with a small panel back then, right? Imagine if you want to do a whole exome sequencing, meaning you have a 20,000 plus genes. That means you need to build 20,000 result components, which is, crazy job, right? You don't want to do that. Again, I came up with some other ideas to get around it, but that's not so much important right, at this point, especially if you're an EPIC user. And also, what are you going to do with the non-exonic regions? And for example, whole genome sequencing, right? And how do you represent the complex variants? Genes with, if a single gene has multiple variants, how are you going to represent that? And also, how can you, represent sequencing metadata like read depth, WAFs, and there are a lot of other things, which are useful for both for clinicians and pathologists to see, to make some decisions on the variant, how reliable the variant is, and so forth. And it's a lot of scrolling, right? It's time consuming to imagine. I showed you this screenshot in the EPIC beaker where we have multiple result components. If you have even 100 components, you go scroll up and down. It's a lot of process. And also manual data entry of genetic variants is also error prone. So anyway, the last aspect here, we were able to automate completely. But again, I will not be discussing this too much for this aspect, but I'm going to discuss more on current practice where, especially for EPIC, where they have the genomic module. So this one, again, now I'm getting into more a latest implementation we done maybe 1.5 year back and stuff. Is again, I'm going to go through the high level here, and then I can get into more details. So this is completely automated process. There is no, I think most of the steps here are completely automated, meaning I'll discuss here. So for example, let's start step by step here. The clinician order tests, in the EHR or EMR here, and then it shows up in the LIS Epic Beaker within the lab that, hey, this is a test has been ordered, and not only that, it shows in the Epic Beaker, but also it generates a HL7 message, a flat file message into a FTP server within the institution. Okay, so the two things happening. One is the LIS is in the LIS system see the message, and also there's a message incoming message order file is also stored on a network drive within the institution. So the lab, obviously, they start do the specimen processing, they do the sequencing. As the sequencing is done, there is a automated batch job we run in the lab, which basically picks up the as soon as there's a triggering mechanism where we know that, okay, as soon as a sequencing is done, upload the data into our high performance computing. Okay, so nowadays you can go around using Illumina has this integrated ICA platform or base-based platform where you can do analysis there, instead of even working on HPC. So once all the bioinformatics is, again, this whole thing is triggered automatically also. All the analysis is done automatically. And just want to let you know, this analysis, the trigger for this analysis is as simple as our bioinformatics, our lab. I'm talking about non-bioinformatics people. The lab molecular technologists, they just use simple Excel to say that this sample is ready and change status to ready or S in the Excel file. And that's all it required. And then as soon as they close Excel file, everything starts here and then gets into this QIAGEN. QIAGEN is, it also uploads the variant file or the raw variants, genetic variants, into this QIAGEN web application, which our pathologists and lab technologists in the molecular lab use for the variant filtering and building the clinical report. So as soon as the report is ready, once the report is ready and finalized by the pathologist, the final report is automatically pushed into, again, the same network drive or the folder where we initially kept our incoming order messages. And we have I'll share my publication recently, just got published a few days back actually. We built this custom HL7 genomic middleware, which takes in these two files and does a conversion using a Python library which we built into outgoing result message, which is then picked up by Epic, the interface engine, and pushes into LIS. And once it's in LIS, it is pathologists will have the two things we can do, right? We can auto, finalize it, but in our case, we did not do auto prelim. We did a prelim, status, and our pathologists or lab technologists, will go one more time and just finalize it, and it goes to EMR. So as you can see, everything is automated here, and again, even this part within LIS could be auto, send out, but we wanted to have at least one review to happen before going to EHR. So anyway, so this is a quite awesome thing, what we did, and then again, this was adapted by many other. At least we are invited to, present this aspect to many other institutions, and also recently at the Epic annual conference, we were invited to present this model. So this has been well appreciated in general, and it worked out very well. And again, now I'm going into the Epic Beaker, right? I was telling you earlier, about Epic came up with its own, native genome management, modules, if you think of it, right? So you should distinguish between LIS and EHR. LIS here, what are the capabilities we have within the Epic Beaker LIS for the genomic data, and then how is it represented once you send into the EHR in the genomic module. Just want to differentiate those things. So this is a screenshot of the Epic Beaker module, right? As you can see here, there is a tumor, so it represents a lot of data here actually. So again, I just want to pause here before I go further. Again, this data is automatically entered into Epic Beaker. This is not entered manually. Just want to letting you know. This is everything was automatically pushed into Epic Beaker, and it's ready for the pathologist or the technician in the lab to just review it. They're not even entering any of this information manually. So you have it, here, all the data. Of course, you see this, different. I don't want to get into all the details here, but you have the specimen level information like a tumor percentage and a barcode and other things, and also the variant level. It's much more detailed, right? You have the the proper annotation of what the full variant is and what type of variant, simple variant or complex and all the things here. And let me go through further here. So if you click on the more details here, you can clearly see under variants, you see that you have the pathogenicity of it, or uncertain. The status is easily found. And when you expand this view, it's going to show you a lot of other things, the gene name, DNA change, amino acid change, and the variant allele frequencies here, and read depth, sequencing read depth. So it captured almost everything what you need, to look at the genomic data here. And there are multiple other areas, also there's a basic information, sequence change, variant coding system, region. These are all the multiple collapse tabs, but if you expand each one of them, I'll show you next slide. So for the basic information, right, you have all this information. Again, I'm not spending too much time on this one. It has been recorded, and also your Epic folks can show some of this information. So it is showing you, variant types and the significance of it, and multiple other things, as you can see in the basic information. And then you have a sequence change at the DNA level, transcript level, amino acid level, and also GVS, and a molecular consequence also. And there is again region information here, which is much more detailed, and also you can have a phenotype information as well for germline testing and so forth. So once that is a Epic LIS view, once you finalize it, it gets into Epic EHR, and this is how a clinician gonna see their results in the EHR. Okay, it's quite beautiful, right? I mean, it's you have much of the information you needed in a very clean approach, and in this case, it addresses a lot of the challenges I discussed before. So this is very nice in overall. But the only thing I would wish here is to have, this collection dates, right? If you're doing multiple testing for the same patient, you would like to see how things changed, in more of a timeline fashion. That is not still here, but there are ways to do that also. But again, we would leave it for other time on that aspect. So the advantages we see with this approach is it is easily accessible to clinicians via result view, searchable for retrospective analysis, clinical decision support could be done. Genomic reinterpretation also could be done for the pharmacogenomics and stuff. They have this reinterpretation thing, a knowledge base there fed into the Epic EHR and Beaker. So it's doable, Epic EHR, so that the decision support could be made, what you call very dynamic manner. And also, this Epic at least started adapting some interoperability standard developed by HL7 Genomics working group. But again, I'm going to get into that little more detail a little later in the slides. But for now, there's some adaptation in interoperability standards, okay? Limitations is, again, can only store limited amount of genomic variants. As I mentioned earlier, right, there is, the EHRs are not meant to design to hold these millions of variants. They only, again, you still store limited amount of actionable variants in the EHR. And again, it cannot store raw sequencing variants, no native genomic interpretation knowledge base. You still have to depend on external knowledge bases, before feeding data into EHR, and no automated clinical decision support. That's meaning a lot of times you should build for each gene, or it's not completely automated. If you think of pharmacogenomics, is a little bit easier example, but if you look at somatic or germline, other germline testing, I think you need to build them one by one. So this is a publication. I think we published just maybe a few weeks back, or less than a week now. It's available online. So the more details about the GitHub, the code and everything what we did is open source. And again, I think when at your institution, if you're adapting something similar, I would be happy to engage with you or help you out with the code and also with understanding, tweaking some of the areas within the code for your institution. So let me switch gears now into so we know that what is the current practice is with the genomic data represented within the LIS and EHR. Let's move on to the 5 genomics data standards and what is the status of it currently. So as I mentioned earlier, I think EHR basically Epic is I think I would say I can call it as the only first and also only EHR which represents, which can represent genomic data natively within the EHR, right? So when they implemented, adapted this, they used HL7 V2 standards, okay? But the newer approach is for interoperability of healthcare data is HL7 FHIR. So there are several advantages of using HL7 FHIR versus HL7 V2. One thing is it is a better data model and a better alignment with the GA4G, which is another organization which does genomic. For example, if you look at VCF files and annotation files, they are the one who set standards. And the second thing is it also the HL7 FHIR supports CRUD operations, and it also better supports the CDS, clinical decision support, and also building smart on FHIR applications on the EHR is pretty easier compared to HL7 V2 messaging. So again, I'm a co-chair for HL7 Genomics Working Group, and we've been working on a lot of HL7 FHIR standards, and that's where things will be going in the future. And we have engagements from Epic and also Oracle. Cerner now became Oracle Health, which was bought out by Oracle a few years back, and other stakeholders in helping out, developing the standards. So there is, again, this is some of these things are new for the community, I guess. So there is there are 2 aspects here. Maybe I can, the next slide I may have it, I guess. Let me see. No, not here. Okay. There are 2 aspects here. One is you have HL7 organization, which sets up the standards, right? They have a multiple working groups. For example, genomics has a working group. There's a lab as a working, different communities within the healthcare has domains within healthcare have their own working group to create standards. But the thing is, there's also something called accelerated programs. Okay, what it does in accelerated programs is there are some standards developed here, right, in the HL7 working group, as an example, and then the accelerator group, what they do is there'll be partnership from companies, private companies, and also academic institutions and EHR vendors and app developers to come together and be able to test those standards to see are these really meeting the requirements, So they do pilots, and real pilots with real, close to be real data, and test out the standards. And if some things are not working out, they're going to give feedback to the HL7 working groups, and they're going to address those. And again, it's iterative cyclic process. So for the genomics work, we have this Codex Fire Accelerator program, under which we developed this GenomeX use case or program under the Fire Accelerator program. Okay, so here we are currently testing out two things. One is Fire Genomic Data Exchange, which is how do you convert your lab report or whatever the lab report data you have into a Fire file or Fire representation of the data, a more electronic representation of the data, and then exchange it with the EHR, or with other institution, however you want to. So that is what being tested out in the EHR Genomic Data Exchange. And then we also have Fire Genomic Operations, where I'm going to talk about this a little more in later, where how can we retrieve the data from the databases, genomic databases. So again, I think if any of you are interested in participating, because one of the complexity of genomics testing is genomics in general is it's a multi-stakeholder thing, right? Developing standards, it requires things were done really isolated. Pathologists, they were doing something their own, and clinicians, they have something on. So I think it's more of a multi-stakeholder thing where clinicians, pathologists, app health informatics app developers, and EHR vendors, all of them need to come together and come up with a standard which is more practical and a viable solution. So I would really encourage any of the faculty members at your institution or from the health IT side, anybody want to participate in these working groups. I think I would really welcome you contacting me, and I can include you in these initiatives. So I was mentioning there are two use cases in the genomics use case, two pilots we're doing under the GenomeX use case. One of the data exchange. The idea here is the orders are placed is HL7 Fire architecture, right? And then the genomic lab testing is done, and they send the results back in the HL7 Fire architecture. And the healthcare organizations, either it can go into EHR or it can get into your genomic repository. So this is we're basically testing the order placement, and then result, sending the result back into the healthcare organization, to the EHR. So next use case, what we're doing is, I know we are almost 10 minutes into the we have 10 more minutes. I'm going to just wrap up in a few minutes now. This is a second pilot we're trying to do is genomic operations work stream or use case or the pilot here. Imagine this, okay? So let's say University of Minnesota, I'm just giving an example. University of Minnesota has their own genomic repository, and nowadays, most of the databases have this API way of calling things, meaning for non-informatics folks, what you do is you just give a URL, with the information. Okay, you give URL with the patient ID equal 123. 123 is the patient ID here, right? And then if you give this URL, if you have access to the database, it automatically pulls up, a flat file with some information in it, for that patient. So if you want to, for example, if you want to have a what you call information for a patient ID, maybe 1001. All you have to do in this RESTful API is just change this into 1001, and you push in the browser, you're just going to show up all this the information related to that patient. So the point is, most of the current databases, have this API way of calling data. It's pretty, standard nowadays. But the challenge is, if University of Minnesota may have a different URL representation to pull the data, and maybe CHLA, for example, second server, genomic server, let's say it's a CHLA, they may have a different URL to call the patient, pull the patient data, and another institution may have their own way of calling the data. And not only just how you call the data is different, but also the result, what you get back from these calls, are also different. So if you look at it, almost all these three of them are doing the same work, right? It is trying to still provide the information about the genetic variant for this patient, but they're represented differently. So you must have already understood the problem here is, it is not standardized. Meaning, let's say if I'm an app developer or somebody, or institution IT who want to exchange the data, I need to build to get this, retrieve this, get this data. I need to build a separate interface or a integration program which knows, for University of Minnesota, this is how I call it, this is how I parse the data, and so forth. And for CHLA, I'm going to do this one. So imagine if you're doing hundreds of institutions, so you should manage the hundreds of mapping information, which is not a viable solution. So what this operations does is it standardizes the API layer, and also the report coming out of the calls also standardized. So that means, as long as I know that University of Minnesota is adopting this FHIR genomic standards, I know what URL to use. I know what did I can get back. So it's pretty, I need not even talk to the people at the institution to get to understand those things. Without even talking to them, I know I can build my application. I can integrate or prepare my application to integrate the data from external institutions. So anyway, so I think I'm gonna push it fast here. So I think I showed you the paper screenshot earlier. That should have the Python libraries and the middleware we created. Again, there could be some tweaks you need to do to adapt to your institution, but it provides good enough framework. The code will already have enough framework inside there. You just need to do some minor tweaks there to implement it to your institution. And again, I think please engage in this what you call genome data standard development process. It's really helpful for all the pathology community, including pathologists, to be there to provide expert knowledge. And I'm going to stop here, and I'm ready to take some questions. Thank you very much.
Thank you, Dr. Chamala. I mean, this was fascinating presentation. We've been kind of working a little bit along the same lines, maybe not as advanced, and focusing mostly on pharmacogenomics. We've actually yesterday tested the first HL7 transaction from pharmacogenomics, going from Go4PGX into the EPIC. And I have Steve Johnson here as well. I'm sure he will have some questions, but I had on the back of my mind also thinking that we should start thinking about taking those experiences and translating those into our somatic testing. And I see Andy is already on, so he will have some comments. So go ahead, Andy.
Thanks, Pawel. Again, Andy Nelson. I'm one of Pawel's colleagues in the molecular diagnostics lab, and I handle a lot of our NGS oncology testing. And yes, Srikar. So we're trying to do some of the similar things. I have a number of questions, but I think the first one that I want to ask is actually about how you've driven that display of discrete information in EPIC for somatically tested variants. Here's one comment, and it leads to the question, which is, I'd say 90% of our oncologists don't have the expertise to dive deeply into 50 variants that are in a report. And I'd say 100% of them don't have the time in clinic. But the biggest use case they come to us and want to know is, okay, I know that this is a BRAF mutated case, but to get them on this clinical trial, I need to know, is MAP2K1 tested? Or, hey, this is the first time I'm seeing the patient. Has KRAS ever been tested? Is there searchable functionality in the molecular module in Beaker in the way that you pipe those discrete variants into it that they can query and say, has KRAS been tested? What is the MAP2K1 status? Something along those lines that would really enable them to quickly understand if a patient's going to match onto a clinical trial?
So the challenge, currently the simple answer is no, right? So oftentimes we have the methodology presented in the report, right? That's where we list all the genes we tested. And oftentimes we only list all the genes tested, but we don't represent whether we found any variant. It may not be actionable at that point, right? I mean, a lot of the clinical trials may not be actionable, right? That means we don't really report back into the EHR. Yep. So that's a challenge in the current system. I already mentioned in one of my challenges section at the end of the EHR presentation, right? So, however, one of the ways to mitigate that is, let's say if you have currently, the way I'm imagining the approach is, they're going to call you up and say, hey, have you done this one? Or can you pull that for
Bingo.
It's a lot of time consuming and dissatisfaction that they feel like, oh my God, I've been asking these guys, they're not responding. They don't understand you're already busy with a lot of other things, right? To sign out. So one of the thing is having one of the paradigm you can take, approach you can take is having an internal database. For example, as simple as CBio portal, right? Sell something there locally. And give access to the clinicians, don't be afraid to give access to them and let them go and check themselves, and it saves time for us as a pathology lab. We don't want to be doing the customer service calls, and especially I can, from my experience, that would be the most dissatisfying thing for the clinicians, because the reality is we would not be responding ASAP, right? It will take some time, and that will be like, all of a sudden it becomes like, why are you guys not responding kind of thing. So I think you can take that approach to mitigate that. But at this point of the time, that is not there. But in the future, I can tell you from my experience with all the Epic and other places, people they're coming up with genomic variant database, which is called JAX server, like a PAX system for the coming with JAX system, where what happens is you store the variants externally somewhere, okay, and EHR will interact directly with that system. And that system can hold all the other non-actionable variants also. So in that sense, in the future, let's say, everything goes well, clinicians can directly access that and get the answers for themselves.
Let me ask one quick question, then I'll turn it over to the other hands that are up. And you predicted it, which is, we've been working with Steve Johnson to try and get all of our oncology data into our local U of M CBio portal instance. Are you familiar with, much as you at Florida had link outs from Epic into an external database, I presume one can create links out?
No, University of Florida don't have external links to the external database. It only have links to the reports. We never had that one.
Got it. Okay. Yeah. Because that would be kind of one of the use cases that we'd like to explore, is a link out from the patient's record to the University of Minnesota CBio portal where its data exists.
Yeah. There's just some challenges we can talk offline, but
thank you.
Thanks, Andy. I see two hands. SChamala iPhone has a hands up, and then Michelle Stoffel. So go ahead.
Oh, hi. Yes, I think that would be me. My name is Zoe SciChev, and the question I have is very, very interesting. I am very impressed with the whole integration. So one of the questions is, how do you are selecting or the candidates of pathogenic variants into the system that is actually visible to the clinicians? And are there any other sort of links that, if that pathogenic variant is related to a disease, that, how is that pathogenic variant like a reference or link that says that, well, this was selected based on, this publications, or how they're graduated to be basically pathogenic variants. So then the clinician can make those decisions. And if you are, so are they related to, because some of the genomic variants are predicted based on, correlation studies, but have you guys also kind of correlated to, proteins are the drivers of functions. So are there any protein data that can be, explored or connected to it?
So I think I would defer that question, because that question is more a molecular pathologist question, right? And how they want to filter things and so forth. But in terms of EHR point of view, that level of discrete information about that will be present in the report also. Again, you have the discrete data, but also you have the report also available within the EHR, which has that information about what is the what you call supporting evidence, or all those things will be there in the report, but not at that much discrete level for the clinician.
Michelle?
Okay, thanks.
Oh, yes. Thank you for this really great talk, and very timely, I would say. Based on, I think others mentioned that, we're currently in the process of kind of going through our HL7
recording
version 2
stopped
build right now. So I guess what I would ask is, we're in this weird spot right now of we know fire is coming. We're really, as a system, hasn't really operationalized much use of this yet. So since we're currently building our HL7 interface for our pharmacogenomics test, what advice would you have to, what can we do now essentially to try to smooth that transition that we know is coming later? I mean,
I think from your side, I don't think much need to be done, because Epic already supporting HL7v2 standards right now for the pharmacogenomics and also for other genomic testing. Epic, again, I don't want to talk for Epic right now, but I think they are part of the table in terms of developing standard. They know the limitation of HL7v2, and they are working towards FHIR. And when that time comes, they will help you with the transition. But there will be some time for it, and I don't think as the institution IT from your side, I don't think you should stress out too much for that right now. And again, there are multiple ways to do it, but I think at this point, nothing is concrete, and they're going to take a slow approach implementing that.
Yeah.
Thanks for this talk.
Thank you again, Srikar, for a fantastic presentation, and thank you for joining us. I want to set up a follow-up meeting with you, and so if anyone is interested, please send me an email, and then we'll set up a smaller group to discuss some of the possible collaborations and how we can learn from Dr. Chamala's experience as we're trying to move our molecular testing into discrete data area in the Epic. And thank you everyone for joining.